Hi, and welcome to Bay Nature Talks. I'm Eric Simmons. I'm the digital editor at Bay Nature magazine. I'm also working on a book about the changing ocean with UC Davis marine scientist Tessa Hill. I'm here this afternoon with two marine biologists and co-directors of citizen science at the California Academy of Sciences in San Francisco, Alison Young and Rebecca Johnson. Hi, and thank you guys for being here.
Hey, Eric, thanks.
Hi, thanks so much for having us.
So I want to just talk about some logistics first. This will be about a 40-minute talk in two parts. We're going to include a lot of really amazing pictures. Rebecca and Alison will talk first sort of about the basics, how to go tide pooling safely, what you can see, and then second, why tide pool observations are and the projects that people who visit the coast can contribute to. Afterward, we should have about 15 minutes to answer questions. So if you have a question at any time during the talk, please use the Zoom Q&A button at the bottom of your screen to type it in. We have a lot of people here. So we will do our best to get to as many questions as we can, but we may not get to all of them. And please note that we are not able to help with technical questions about Zoom in the Q&A. So we'll record the webinar, email a link afterward. If you are having technical issues, it'll be viewable later. And then finally, for those who haven't seen us, Bay Nature magazine is a nonprofit magazine and website. You can help us keep writing and reporting on the natural world by subscribing or donating at baynature.org. And we thank especially those who made the suggested donation for this event tonight. Your support lets us bring you talks like these. Now to tide pools. People have looked into the pools of water at the edge of the sea for as long as we have been a species. There's evidence that our human ancestors were traveling to the coast at low tide, gathering mussels and sea snails dating back to 165,000 years ago. Tide pools, in part because of our long relationship with them, have become one of the best understood of all marine environments. And they are now where we can go to watch changes in the global oceans play out in microcosm. We have today two great interpreters of the tide pools here to talk with us about how to go tide pooling, what to look for, and why it matters. Northern California and the west coast of North America generally have some of the most famously diverse tide pools on the planet. These are in some instances comparable in diversity to tropical rainforests or coral reefs. Rebecca and Alison have spent decades studying these environments, and even better, bringing thousands of amateur naturalists along with them to learn to see this world. With great low tides happening this week, there's an opportunity right now to explore for yourself and to participate in intertidal science. We do want to remind everyone, of course, to follow COVID guidelines and to be safe. A lot of popular tide pooling spots, including Fitzgerald Marine Reserve in San Mateo County, are fully closed right now. So please do check before you go. And we've also heard from a few readers they're concerned about encouraging people to visit the natural world right now, given crowding and the potential for disturbing natural resources. We feel, however, this is not a reason to tell people not to go, but rather an even stronger reminder of the value of helping people to visit respectfully. I want to acknowledge, too, that we are talking a lot about beauty and diversity of natural resources, but these are also cultural resources. West Coast tide pools have been visited, stewarded, and appreciated by indigenous groups for millennia. In the Bay Area, where so many of us are today, this means we appreciate the connection to these places of the Rumsen, the Amamutsun, the Ramaytush Muwekma Ohlone, the Coast Miwok and Federated Indians of Great Rancheria, and the Kashiapomo. Okay, so let's learn about these special places. You want to visit a tide pool? How do you do it? I'm going to turn it over to Rebecca and to Alison.
Awesome, thanks so much, Eric. Alison's going to share her screen, and I will start first, and then I'll hand it off to her. So the tide pools, right? I mean, this photo kind of really says it all for me. So this is not from California. This is a photo that Alison took in British Columbia. But, you know, I just really wish I was here, this place right now. And I think we all can recognize the beauty and the peacefulness and just the diversity and the amazing, just the intriguingness of places like the tide pools. So next slide. And so when we think of tide pools, I mean, in my tide pool dreams, right? It's a place like this. This is from the Sea Ranch up in Sonoma County, and Alison took this picture of me, which is one of my favorite pictures of all time. Because it really captures for me that one of the things I love about tide pools, they're just simply magical, right? So they're the places where the land meets the sea, where seaweeds and animals thrive, an amazing place of diversity. And like Eric said, they've been so important to humans for millennia. And most recently, for Western science, they've been a place that we've really explored a lot of basic ecological principles. But you can only get there certain times. So they're also they're mysterious and magical. So you can't just walk it out at any moment and be in a place like this. So next slide, Alison. I want to show you a little bit about the place where Alison and I do most of our work, which is down at Pillar Point in San Mateo County. So just to give you a little orientation, you can't see my slide, but maybe Alison can kind of circle where Pillar Point is on this map. So next slide, just south of San Francisco, about 45 minutes in San Mateo County. There's this little headland that sticks out. It's called Pillar Point. It's most famous for the Maverick Surf Contest. Next slide. You can see here, it's even labeled Mavericks Beach, destination for big wave surfers. But it's really some of the best, I think, tide pooling close to San Francisco. And if you look at this picture, you can see it's looks like a beach, right? Here's the sandy beach, because this is a picture that's taken at high tide. So you can't see the rocky reef at all where we would tide pool, where we would see all these seaweeds and the diversity of life. So next slide. But this is what Pillar Point looks like at low tide. So you can see this huge expanse of rocky reef or rocky coast that's exposed at the low tide. And this is when we want to go to explore the tide pools. Next slide. This is another picture from the beach at Pillar Point. This is at the high tide. You can see these sail rocks called sail rocks far offshore. And then next slide. This is the low tide, right? So you could come, depending on when you arrive at this beach, it looks really, really different. And you can see all these creatures that you're going to hear about at a low tide, but you would see amazing birds and amazing things on the beach when the tide is high, but it's quite different. So you have to know a little bit about when to go to be able to explore. So next slide. And we're going to show you some amazing pictures of super, super diverse places that look like they're really far from cities, really far from people live. But I want to let all of you know that you can go tide pooling lots of different places. And it doesn't have to be somewhere that you've read about in a book or that is highlighted as the best tide pooling space of all of California. There are lots of little pocket beaches. Anywhere that you can find rocks really or hard substrate on the shore is a place that you can find seaweeds and animals that are thriving. So this is at Muir Beach, just north of San Francisco. So next slide. Just at the south end of Muir Beach, you can see a lot of diversity and you can stand on the sand, which sometimes is great because you're not standing on any of the animals or seaweeds and it can be a little more accessible. Okay, next slide. You can also tide pool in San Francisco, not just on the rocky coast, but this is Aquatic Park near Fisherman's Wharf, between Fisherman's Wharf and Fort Mason in San Francisco. And next slide. There's a lot of riprap and things that have been put in the ocean to protect from erosion or just dumped there. And you can turn these rocks over and see a lot of amazing creatures that Alison will talk about later. So next slide. And you can even go on the bay side. So this is from Heron's Head Park. You can tide pool in San Francisco Bay, and you can see a lot of the things, and even some things that you can only see in San Francisco Bay. Next slide, that you can't see anywhere else. And these are all really amazing places to explore. So although most of the pictures we're going to show you today are from not these places, I just want to let you know that you can walk maybe from where you live or take a bus when it's safe or drive just with your family. And you can explore the tide pools everywhere. All right, next slide. All right, so let's talk a little bit about how to go to the tide pools. How do you know where to go? How do you know when to go? How do you know what to wear, what to bring, what the rules are? So I'm going to go over some of the basics. All right, next. Oh, but first, I forgot. I wanted to show this quote. This is one of my favorite quotes of all time. This is from John Steinbeck in The Log from the Sea of Cortez. And for those people, those of us who study tide pools, we see this all the time, but it's really, really an amazing thing to think about. So when you go to the tide pools and you look carefully, you can just see so many different things and so much diversity, and it makes you kind of feel small and big at the same time. This is what it means to me. So you're looking at the tide pools and you look really carefully at small things and you look up to the stars and think about big things. It's a really nice place to kind of contemplate our place in the universe. And it's amazing. So next slide. Okay, the first thing right now, if you want to go tide pooling, where do you go? So the first most important thing is to check on your beach. Like, is it open? Can you go? And after this webinar, we'll send you a list of resources. So a lot of the things that I'm showing here, we'll send you links But there are a couple of great places online. The Chronicle has this website that tells you what's open and what's closed in the Bay Area. This is not just for beaches, it's for parks as well. So next slide. The LA Times also has a tracker of beach closures for county by county for the whole state. And next slide. And Together Bay Area, along with I think Post or MidPen, apologize if I'm mixing you guys up, has a website that's Bay Area Outdoors that talks about everything that's opened right now outdoors. So if you're trying to figure out where to go, you can look here and see if the place that you're thinking about is open, or maybe find somewhere to go that is open. Okay, next. And then check the tides. You need to go tide pooling when the tide is low. So there are a couple ways to do this. One is to go to the Google NOAA tides, find the place nearest you and find when the tide is low. If you still get a paper newspaper, you can look on the weather page and you can find out when the tides are low for like the 3 days around the day that you're on. Or you can use the tide book. And this is just some shots of next week's tides from our favorite tide book, the tide log. So right now the tides are low really early in the morning. So you have to be ready to get up early. Next week, I think the low tides are around 6 a.m. So you got to be ready to get up early, but make sure you check. We like to arrive kind of before the low tide, so a couple hours or an hour before the extreme low tide. You also need to know the rules. So check to see if you're going to a marine protected area. This is a map of marine protected areas, so you can find out about the rules about where there are not, what you can and can't do inside MPAs, the marine protected areas. All right, next slide. If you are thinking about fishing or harvesting anything, first of all, check if you can do that in the MPA. Second of all, read the ocean sport fish regulations to see what you can and can't do. And you would need a fishing license. So everyone needs a fishing license who's fishing. All right, next slide. And then when you arrive at the beach, make sure you follow the local rules, because beaches have different local rules. So this is a sign from Duxbury Reef up in Marin County, and they have their local rules posted here. All right, next. Next, what to wear and what to bring. So wear boots or other kinds of shoes that can get wet and have tread. So next, so boots. Next slide. Sometimes people like to wear water shoes and some people like to wear old sneakers. Just have some tread and make sure they can get wet. Right, next. And wear layers, because it can be cold. Even though it's summer along our coast, it can be cold, so be prepared for any weather. Next. And bring a phone to take some pictures. Or next, or a waterproof camera to take pictures of the things that you see. Next. All right, so how do you keep yourself and the environment safe? So I'll go through some basic, really quick rules. Some are just beach rules and some are tide pooling rules. Be mindful of the waves, never turn your back on the ocean. Be aware of deep channels that you might have crossed as the tide is going out, and that you will have to cross again as the tide comes in, because they'll get deeper and deeper and deeper, so just be mindful. Try to pick bare spots to step so you're not stepping on animals or seaweeds, partly to protect the animals, partly to protect you so you don't slip on slippery algae. Try to keep your steps up and down. Don't scuff your feet, because that can harm animals, and make sure your step is secure before you shift your weight. And you don't have to go really far, and you don't have to travel across the whole reef. Find a pool that looks good, stay still, and look really closely and pay attention. Take most of your time to just watch and see what happens, and that's how you really discover the most amazing things. And this is probably the most important, so always follow local rules when it comes to touching animals or touching seaweeds or picking up tide pool life or lifting rocks. Most of the animals that you see should not be picked up because they're attached to the rock and shouldn't be moved. So next bullet, Alison. Animals clinging to the rocks can be really hurt if you lift them off. So really there are very few things that you really need to pick up. If you do want to touch something and it's allowed locally, just make sure your hands are wet and do it super, super gently. Next slide. And of course, if you do lift up any rocks or move anything, make sure to put everything back how you found it. Next. All right, and just get out there and explore. And those are some of the rules and the basics really fast, but we'll send you some links so you can delve into the resources as much as possible on your own time. Now it's up to Alison.
Thanks so much, Rebecca. So yeah, Rebecca kind of gave the overview of how to figure out where to go and then what to do when you're there. I'm going to talk a little bit more about what you should be looking for when you get out to the tide pools. So as Rebecca talked about, you know, the tide pools have been very well studied for years and years. So like, what makes them so interesting and why are they so diverse? And a lot of it is because there's multiple habitats squished into a very small space in the tide pools. You can imagine like walking from the top of a mountain down to the base and then like out to the ocean, and how many different habitats and zones you would pass through. You can do that all within the span of, you know, 100 feet in the tide pools, you know, pass through these multiple zones. And so when we talk about tide pools, one of the first things you learn when you're studying the intertidal is about zonation in the tide pools itself. And so this slide shows from the splash zone down to the subtidal area, what they call the upper, middle, and lower shore, we generally call the high, mid, and low intertidal. But the reason there's these different zones is basically because of the tides, the fact that, you know, the tides are coming in and out for us twice a day, and that they're uneven during those in and out times as well, during high tide and during low tide. And so organisms that live in the high intertidal, they have to be able to deal with lots of stress around, you know, being exposed to the air, having temperature fluctuations. If it's high tide in the middle of a day on a really hot day, they have to be able to withstand warm temperatures. If they're in a little pool, their little pool starts to evaporate, so they have to be able to deal with salinity and definite amounts of sunlight. And so in the high intertidal, you'll notice that it's not covered with organisms because there's only a few types of organisms that can survive all those stressors. But as you move from the high intertidal down to the low intertidal, those sorts of stressors kind of disappear, and instead the main driver is competition. That as you walk down lower in the intertidal, you're going to notice that the space is totally covered with creatures that are all trying to vie, you know, for their little corner of the tide pools. And so as you move into the low intertidal, competition is one of the big factors of what drives diversity and why you see the things that you see down there. So when you go out to the tide pools, sometimes you can see zonation really clearly. Like here, this is again up on Vancouver Island in British Columbia. If you find a spot that has, you know, really obvious topography like that, you can see the zones really easily. But most of the time, those tide pool zones are not as clear as that previous drawing makes them look. In the Bay Area, you can find lots of different types of rocky intertidal areas. Boulder fields make up a lot of the rocky intertidal from Sonoma northward. And so when you look at something like this, you actually have lots of different splash zones and high intertidals and low intertidals. Each little boulder can have its own zonation on it. And so as you look through this area, it's not just about walking from the beach down to the ocean. It's actually looking at all those different rocks and seeing those zones. South of us, like in Santa Cruz and farther south, you often see these really flat bench intertidal where there's hardly any topography at all. And really what drives the zonation is how close you are to the ocean versus how close you are to the beach. And just walking that is how you're going to pass through the zones. And then here in the Bay Area, especially at Pillar Point, which is where this photo was taken, we have a lot of these kind of varied benches that they are these flat benches, but there is a little bit more topography to them. And so a lot of the photos that we're showing today come from areas like this, especially along the San Mateo coastline. So when Eric asked me to talk about the diversity of things you can see in the tide pools, I just want to make it really clear that I'm not even like scratching the surface of diversity of the tide pools. That, you know, if you're out there during a good low tide and you can spend 3 hours out in the tide pools, you can easily see over 300 different species. As you walk in from the high intertidal to the low intertidal and you look really carefully, you can find so many things. But what I'm going to show you is kind of the main things that you can find, kind of the easier things to find, and some of the species that kind of dominate the high and the mid and the low intertidal as you explore the tide pools. So starting in the high intertidal, again, like I said, the high intertidal is not covered in organisms because there are very few organisms that actually can survive all those stresses. But when you do start to look up on the higher intertidal, you're going to see a lot of these small brown barnacles, and there's a few species of limpets especially that can tolerate being out of the water for so long during the day. These little tiny snails called periwinkles, they definitely are dotted throughout the high intertidal also. And then the main algae you're likely to see is this little rockweed. And you can actually see in this photo, this little rockweed has those little periwinkle snails on it and is surrounded by those barnacles as well. So a lot of algae needs to live lower in the intertidal, but little rockweed can do pretty well in the higher intertidal. One of the first ways you're going to know that you're in the mid-intertidal is mussel beds. They kind of are the key way of knowing you're at the mid-intertidal. And for us, that's the California mussel. We have large beds of these mussels that form in the mid-intertidal. And often in the beds themselves, you'll find these clumps of gooseneck barnacles that look really different from those high intertidal ones, or the ones you can see on the shells of those mussels there. They're kind of scaly and have these kind of long stalks that they're on as well. In the mid-intertidal, depending on where you are and the time of day too, you might also find a bunch of ochre stars. Mussels tend to be the very favorite food of ochre stars. And so sometimes you will find them up in the mussel beds or directly below the mussel bed. I will say that a lot of people use kind of that mussel bed ochre star line to delineate the mid-intertidal from the low intertidal. So sometimes ochre stars are often definitely in the low intertidal as well, but they will move throughout the day and with the tides to go up into the mid-intertidal to eat those mussels too. So I just wanted to point them out. That's why I put them in the mid-intertidal, because you could find them there, but they're definitely in the low intertidal also. As you explore kind of the nooks and crannies and the rocks of the mid-intertidal, you're definitely likely to see some shore crabs. The two main ones that we find in our Bay Area tide pools are the striped shore crab and the purple shore crab. The mid-intertidal is also a great place to see chitons. You can see chitons from the high, kind of lower high, all the way down to the low intertidal, but the mid-intertidal tends to be where they're bigger and where they're a little more obvious. And especially those mossy chitons, the ones on the lower left there, they tend to be the most obvious and kind of most dominant chiton species that we see in our tide pools. But as you look throughout the rocks, you're definitely gonna see lots of different species of chitons. Aggregating anemones, especially closed ones, if you're there at a nice low tide, most aggregating anemones in the mid-intertidal will be closed. You can see on the right that those ones are pretty obviously anemones that are just closed up. On the left. On the right, though, you can see these little aggregating anemones sometimes cover themselves in like rocks and shell bits and sand to actually help them keep their water inside so they're not desiccating during the low tide as well. So sometimes they're a little harder to notice. Sometimes you might not notice until you step on them and suddenly the squish of water comes out. But once you see them, you'll be able to recognize them and you can avoid them. And when we look at the algae in the mid-intertidal, there tends to be kind of what we call a red algae turf, where there's actually lots and lots of species of red algae that grow in the mid-intertidal, but they tend to kind of be shorter and just kind of form this turf layer throughout the mid-intertidal. And finally, venturing down to the low intertidal, this is a great place to see open anemones. There's quite a few species of anemones that we have at our tide pools, but these three species are probably the most noticeable. There's the giant green, which is right there in the middle, the sunburst anemone that's kind of off to the left there with the stripes on the inside of it. And then you can see some open aggregating anemones kind of along the bottom of that photo there. Again, like I said, ochre stars are definitely in the low intertidal as well. So as you're checking out those anemones, you might find a scene like this where you're seeing anemones and ochre stars. But more commonly, these two stars definitely tend to hang out more in the low intertidal. They don't move as much as the ochre stars do. So bat stars and leather stars are two different types of sea stars to look for in the low intertidal in our tide pools. I could have made this entire talk about nudibranchs. We have so many amazing nudibranch species along our stretch of coastline, but I decided just to show you 7 that are either pretty common, so you can find them, or they're nice and brightly colored and they're big. So these are the ones that I would say you're most likely to find as you venture out there. They're bright yellow or orange or pink or electric blue. And they tend to be, these are some of the bigger ones that we have out. But if you can find a nice big pool in the low intertidal, this is a great place to do what Rebecca said, which is just to sit and watch. And eventually you might see these guys kind of cruising throughout the red algae in the low intertidal. Lots of different snails in the low intertidal as well. The ones on the left, these tegula snails, especially the black tegula, they actually are pretty cosmopolitan. You can find them kind of from the low, high intertidal all the way down to the low intertidal. Those brown tegulas are more kind of mid to low intertidal, but they're definitely kind of throughout the tide pools. These 2 top snails on the right are definitely more of a low intertidal species. And again, these are ones that you would find in a pool in the low intertidal. Urchins, you might find a big urchin bed like this where they've actually carved those little holes themselves that they like to sit in, their little homes. Or you might find urchins kind of just scattered throughout other organisms in the low intertidal too. It really kind of depends on the type of rock in the place that you're going to, and also how much space is available for them to make those little holes. As you approach a tide pool, you might see something like dart away and hide in the rocks. Those are very likely sculpins. My one call out to vertebrates in my slides here. There are actually quite a few different species of sculpins in the tide pools, and it's a little confusing because there is a species called the tidepool sculpin, but it's not the only one you find in the tide pools. And they can be amazingly camouflaged on their background as well, as you can see in these 3 photos here. And then finally, when you look at the algae, you're going to find some larger red algae in the low intertidal. You can see on the photo on the left, kind of interspersed with some kelp, or you might find just large masses of kelp in the low intertidal as well. But that is our quick walkthrough of the diversity in the tide pools. So now I'm going to turn it back over to Eric.
Thanks, Alison. This is great. We already have a question. Can you just do a future talk about nudibranchs? So we've done our job. Yeah.
Yeah.
So thank you, Alison. Thanks, Rebecca. I hope everybody watching has a new goal now for something to go find out there and for something to look for. I wanted to just transition now to talking about, taking these observations that we can make and turning them into a better understanding of where we live and why these places matter. I do want to say just a reminder first, if you do have any questions, enter them anytime in the Q&A field. We'll go through them in the last 15 minutes or so. So as Rebecca and Alison have mentioned, these places are so diverse, they're very small, they're a part of the ocean we can visit easily and regularly. So we can learn quite a bit from them. One of my favorite stories is, the term keystone predator, coined originally for the way those ochre sea stars behave in tide pools. And that, we can use that term for wolves or otters is in part because we can see it happening in the smaller scale. And these are also places that change fast. They reset twice a day, more or less, with the tide, and we can see them. So what I really am excited to hear about now is from both Alison and Rebecca, a bit more about how we can learn about these places, what we can learn, and how it can better inform our understanding of this place where we live. So I will turn it back over to Alison.
All right, let me share my screen again. There we go. Back to that beautiful sunset at Pillar Point. Yeah, so Rebecca and I, although we are tide pool connoisseurs and we spend lots of time in the tide pools just to enjoy them and to see what's out there, we do also study tide pools. That is part of our job at the California Academy of Sciences. But we also run the Citizen Science Program, where we get people involved in doing research with us for the Academy. And we do this in lots of different ways, but because we're both marine biologists and because we both love the tide pool so much, and we started our Citizen Science Program, we really wanted to have a way to get people out to the tide pools and then to actually do some research out on the coast itself. And so one of the reasons that we spend so much time at Pillar Point and we have so many photos at Pillar Point is that we have some long-term monitoring that we do at Pillar Point, but we do it with volunteers. We've been working at Pillar Point since 2012, so about 8 years now. And the reason we started working here initially is that directly adjacent to Pillar Point, when California was designing its state marine protected areas, they put a marine protected area directly adjacent to Pillar Point, but actually left Pillar Point outside of the marine protected area, basically to serve as a control, so they could monitor through time what's the difference within a marine protected area and right outside of one. But at that point, there wasn't even like an available species list for Pillar Point. So Rebecca and I, when we were designing our citizen science program right around that same time, we thought this would be a great place for us to step in, get volunteers and interested folks out to the tide pools and help us gather data that we can use to better understand what's going on at Pillar Point through time. So one of the things that we do is we have 6 permanent plots along the edge of the Pillar Point Reef that we monitor every year. We are usually out there 2 times a month. About twice a month we have a good low tide series, so we try to go out for both of those low tides, and we try to monitor each one of these plots 2 times a year. So that's at least 12 times that we're out there with volunteers throughout a given year. And what we're doing in these plots is we're counting organisms, a kind of particular group of organisms that we've set up. And I'm going to show you a little video about what this looks like. If it will play. There we go. Where we're working in 10-meter radius plots. This is the day that we had 2 volunteers out there with us, because we can do it with almost any size number of people. No one needs to have special training to come work with us. All you have to do is be able to kind of search tide pools and look for things and call us over if you don't know what they are. And we count things like nudibranchs and sea stars and urchins, species associated with mussel beds and things like that as well. And this is greatly sped up. It usually takes us about a half an hour to monitor one plot while we're out there at the tide pools. But one of the other things that we wanted to do, besides setting up these plots where we're actually kind of counting things and using a data sheet, is we wanted to build a species list, and actually more like a species atlas for Pillar Point. We wanted to understand across the entire reef, not just in those 6 permanent plots, what are the species on the reef, what are the common species and what are the rare species, and where are they showing up on the reef itself. And so what we always do when we go out and monitor our plots is we also spend some time with our volunteers literally just tide pooling and searching the area. But what we do is we take photos of the things that we find and we upload them to iNaturalist, so we can keep track. Every time we're out there, we're taking photos of things and saying these species were here today and here's all the places that they were. We're not going to go super in depth to iNaturalist. You should definitely check it out if you're unfamiliar with it, but a quick little how it works is that if you're using the app or if you're using an underwater camera, these instructions are a little bit more for the app itself. All you have to do is take a clear photo of the thing that you're looking at. You don't have to know what it is. iNaturalist will actually give you a suggestion of what it actually thinks it might be, the identification of what species it is. You can pick from that list, or you can type in your own ID if you know what it is, or if you don't agree with any of those options. And then you share to upload it to iNaturalist. So to kind of show you step by step what that looks like, there's that exact same photo before of that little yellow nudibranch. Here's what it looks like in the app itself. You can see because we're using the app, it actually records where we are and what day it is. So it lets us know, the location and the date that this thing was found. When you click on the view suggestions, iNaturalist brings up its suggested list of species based on what it looks like, and also tells you what's also been seen nearby in that exact same area. So you can see they're saying it's pretty sure it's in the genus Doris in their top 10 suggestions. You can only see the first 3 there, but you can see they're all yellow nudibranchs in those first 3. But the very first one that it suggests, the Monterey Dorid, actually is the correct identification. And so you can pick that and assign it to your observation, and then you upload it to iNaturalist. And when it gets to iNaturalist, the power of iNaturalist is the fact that it's a social network. It's made up of a huge community of people that are not only sharing their observations, but they're also helping each other identify what they saw, so that when that nudibranch photo gets uploaded, the community can say like, yes, that's the right ID, or they can say, oh, it actually is this other one that looks like it. Here's why we know. Or if I just uploaded it and just said that it was a nudibranch, it could tell it, the community can help me narrow it down to what species it is. So that's really the power, is that once you upload your photo and then everyone can see it. So you can see here's that same observation. This is now basically taking this photo and turning it into a data point, because now we have where it was seen, we have what day it was seen on. I'm the one who saw it. And then the community is agreeing that yes, this is a Monterey Dorid, which makes this what we call a research grade observation. So what was just a photo of a nudibranch is now actually a data point that says a Monterey Dorid occurred at Pillar Point on February 28th, 2018. And now that's part of a species occurrence record that can be used for lots of different things. Those observations are on the huge iNaturalist database that has almost 45 million observations around the world so far. A lot of people like Rebecca and I use iNaturalist too to create projects to help us kind of aggregate the data that we're the most interested in. So that observation not only is on iNaturalist for anyone to look at and to use for their research purposes, but it also gets added to our Pillar Point project. And so you can see we now have almost 24,000 observations that have been made at Pillar Point, and we found over 650 species in the years that we've been out there, that people have been documenting the things out there. So that's a way that we can basically take a photo and turn it into a data point. And this is just a few of those 650 species. So now we have this amazing resource that tells us what species have been found at Pillar Point, how often they've been found at Pillar Point, and also where on the reef they've been found as well. So we really have created a species atlas, and all it does is you'll have to start with is just taking a photo and then uploading it to iNaturalist. So I'm going to turn it over to Rebecca to talk a little bit more about how you can do this work that we do at Pillar Point, but anywhere along the coastline.
Awesome. Thanks so much, Alison. So, as Alison mentioned, we have been doing work for a long time at Pillar Point, and while we were there and are there regularly, like every couple times a month for now 8 years, there are a lot of changes that we've noticed. And so I'm going to talk about a couple of those changes I'm only going to highlight 2 today, but there are actually lots of things that we've noticed change and lots of data that we've collected to help us understand the changes that we're seeing in one place at Pillar Point. But we realize, of course, that we're just getting a lot of data from one place along the coast. But California and the west coast of North America is a really big place. And so we developed this program called Snapshot Cal Coast in 2016 to work with communities up and down the entire California coast to do what we do at Pillar Point, but just do it at one point in the year, so we can look at what's happening every summer and get a snapshot of coastal biodiversity at one time and compare that year over the year to understand the changes that we're seeing. So next. So one of the changes that we saw, and I saw someone mention this in the Q&A, so it was the starfish wasting disease. So we, in 2013, 2014, we started to see a lot of starfish get sick from this wasting disease and literally disappear before our eyes. So we saw this in our permanent monitoring plots, and we also saw this in our iNaturalist data, along with lots and lots of other scientists along the coast and lots of other citizen scientists. But we were able to see this in person, and we've also been able to see the recovery of many starfish. So not all species have recovered fully, but we've been able to see the recovery. And we could see this at Pillar Point, and we were wondering if you could look at iNaturalist data, if we collected it more regularly, if you could see this same trend using community collected data, not scientists out there with transects and graduate students. So could we see this trend with community collected data? So next. Another thing that we saw is this beautiful nudibranch that Alison already showed you, which is regularly found off our coast, but in pretty low numbers. And this is called the Hopkins rose, and this blooms during warm water events, the little play on words with bloom and rose, usually during El Nino events. But in 2014, during an unusual warm water event we had off our coast called the warm blob, this became the most common nudibranch we saw at Pillar Point. And so these are kind of 2 of the things that motivated us to start this program, to help motivate other people to collect more data using iNaturalist to see if we could detect changes along our coast, the whole entire coast. So next. So every year since 2016, for the first couple weeks in June, we've worked together with amazing partners along the entire coast with support from the California Ocean Protection Council and also Resources Legacy for this work. We've gotten kind of armies of people in small little groups being safe and following the rules like I described, not like a big army, making and sharing observations to help us look for changes in species ranges, to understand patterns inside and outside of California MPAs, and to really understand how this volunteer and community collected data can be used for MPA and coastal management overall. And we're just now starting to look at these data and think about how these can be used as an early warning system for changes along our coast. So next slide. So this work wouldn't be possible without tons and tons of partners, including the Marine Protected Area Collaborative Network. And then this is just a handful of the partners that have worked on this with us. And what Alison and I do is we train and we give tools to use iNaturalist, to mobilize your community to get out to the coast and make and share observations. And we train a bunch of people to do this work with their local folks and in ways that are meaningful and important and feel good for them. And we don't tell them exactly what to do. We say, hey, we just want to collect some data, and how does that work best for you? So next slide. So next. So we really get out there. Actually, let's just skip this one, Alison. So this is like people getting out to the beach and collecting data, and a lot of people doing all this work together over time. Looks like this, where this is the last 4 years of Snapshot Cal Coast. So you can see this small bar at the bottom, we made about 7,000 observations altogether, but last year we made closer to 40,000 observations up and down our coast, and about 4,000 people overall have worked to collect data that we use in this project to understand how ranges and things are changing along our coast. Now this year, it's a little bit different because we are not getting people together to do and make and share observations together. Next slide. What we're asking people to do is go out on their own during low tides with their families or the people that they live with, and just make and share observations any low tide they can between June and November. And so far, people have made actually close to 50,000 observations. So next slide, because it's a longer period of time, right? So we're gonna have to look back at these data, but people right now, if you go out and make and share an observation along the coast, it counts for Snapshot CalCoast. So next. And really what we've done with these data with the support of the Ocean Protection Council is we have a data scientist on our team, Gio Rappucciolo, who's looking at these data and trying to find these patterns, things that we know happened, but can we use these community collected data to figure out, to kind of backcast? And then once we know these community collected data picked up trends that we know happen, how can we then next turn that to look forward and make an early warning system? And anybody can look at this Dynamic Observatory of Biodiversity. It's on the web, and we'll send that link as well. Next. And you can just see some of these things I talked about. You can see this is for the ochre sea star. You can see this decline in ochre sea stars that was starfish wasting. And then you can see the slight recovery of starfish. Next. And then you can also see this huge increase in Hopkins roses during the warm water blob, and then this decrease back to kind of more normal times. So next slide. So this is a link to that website. And so the thing that's most exciting for us about this project is that we're using data that people collected so they could learn more about the tide pools or learn what that anemone was. Is that thing a plant, or is it a more related to jellyfish? Like, people are trying to learn about the natural world and being curious and discovering. And we're taking all those data together and helping protect and manage our coast by working together with the state of California. So no matter why someone shared their observation, because they knew they really wanted to help manage the coast, or because they were just curious, all these data together can really help us understand what's happening along our coast. And we really need people everywhere making and sharing these observations. All right, so that's it, I think, for us. And we have enough left. We packed a lot in there, but we have time to have some questions.
Thank you both again. It's just, it's so much cool stuff to talk about. And I think the questions we've got are really reflective. We've got some great questions coming in, reflecting, I think, so many of the different fun things to talk about. So I thought maybe I'd start with a good one from Roz Whitesides. What makes a good low tide? How low does a tide have to be to go tide pooling?
Yeah, I saw that right away and I was like, oh, I should have said that. Yeah. So, you know, the best low tides are negative. So we can have a whole lecture about tides too, so we won't today, but if you look in a tide book and it's 0 or negative, those are the best times to go. But really, most places along our coast, if it's like ½ foot or lower, you could see some tide pools, maybe even 1 foot or lower. If you really want to see a bunch of nudibranchs like Alison showed, it's going to have to be a negative tide. But yeah, look for that 0. I would say that's a good benchmark.
Alison, anything to add?
No, it kind of also depends on where you go to. As you try different spots, you're going to learn what's the best times, like how low of a low tide you need to go to really kind of explore particular areas. And yeah, like Rebecca said, some places like it can be a slightly positive low tide and it's still totally fine. You can at least see the high and mid intertidal stuff. But yeah, to really get out there and see things like nudibranchs, in a lot of places you do need a negative low tide. And about the most negative we get is usually about a negative 1.8 or 1.9. Usually in the summer is when we get those good low tides. And then again, kind of at the late November, early December is when we get the lowest low tides along our section of coast.
And also, I just recently learned this actually, but if you pay attention to weather, if there's a high pressure system off the coast, the tide is even lower. So imagine high pressure is like pressing down on the water. So it's further out. And so that can actually improve the tide pooling experience.
We have a lot of questions coming in about just how to participate. Is there a way to volunteer with the Academy or with other projects? How do you participate in Snapshot?
Yeah, so if you're interested in like coming out with us at Pillar Point, the best thing to do is if you go to our website, you can go to the calacademy.org and just in the search bar type citizen science. And we have an email link that you can just email us and say like, hey, I want to be on your list. And like I said, you don't need any training to come out to Pillar Point with us. All that does, if you email us our citizen science account, we'll just put you on the list that we send out to everybody. And we say like, hey, here's the next like 3 times we're going out, just come if you can. And then all the training and help that you need, we can give it to you when you're out there as well. I'll turn it over to Rebecca to talk about how to participate in Snapshot CalCoast.
Yeah, I mean, if it was a normal Snapshot CalCoast year, I would be like pushing, saying, come out with us, we're all going tide pooling. But alas, if you go out tide pooling anywhere along the coast of California or in the San Francisco Bay or in the Monterey Bay, into Elkhorn Slough, and you take pictures of the plants and animals that you see and you upload them to iNaturalist, they automatically are added to the Snapshot CalCoast project. So you will be participating in Snapshot CalCoast. You won't be with a whole group of people, but you'll still be doing the same thing kind of dispersed with your colleagues who are out there making observations. And there's a Snapshot CalCoast project on iNaturalist that we can include the link when we send out this list of resources, so you can follow along. You can also help identify finds if you know what the things that people are seeing.
Yeah, and to answer a quick question that I saw in the Q&A too, we're not currently taking people out to Pillar Point. Unfortunately, The last time we were out there was early March. But as soon as we can, we will, we're actually almost to the point where all of our low tides are in the middle of the night. For us, that happened the month of September and the month of October. Our low tides are all happening in the middle of the night and we don't go out anyways. So hopefully by late October and early November, we can start having people join us again. So that's plenty of time to get your name on our email list, and we will send emails out when we start to have people come out with us again.
Yeah, and we're hoping to have a more traditional Snapshot CalCoast the first couple of weeks of December. So TBD based on how we get this virus under control. But so stay tuned. If you add your name to that list that Alison mentioned, or to our Citizen Science newsletter, you will keep you updated.
I'm going to ask you guys two questions. One, because I want you to have a minute to think about the second part. So somebody asked, and in fact, Kate Montana, who you guys may know, asked,
Hi, Kate.
Hi, Kate.
If you have a favorite tide pooling story or moment, which I'm going to further constrain and say in California. And before that, somebody asked a great question: is an anemone a plant or an animal?
So I can answer that one. So sea anemones are animals. They are most closely related to corals and jellyfish. They're cnidarians. They have stinging cells. They don't move very far, although they do actually move, and they can move. So they can look a little bit like plants because they have these like tentacles. But if you look at a lot of those pictures that Alison showed, they have like a little mouth in the center, and they are stuck to the rocks, and they collect their food through their little mouth. A lot of people confuse sea anemones and sea urchins, which is I think it's like they're really close in our brains. So sea urchins are echinoderms, and they're the purple ones with the spikes, but like they both have spiky things. So I hope I didn't make a lot of people now confuse them, but sea anemones are definitely animals. Some of the most primitive animals out there. So.
All right, so how about those favorite moments?
Oh, that's really hard. You know, we've had so many. I really love when we transition from afternoon low tides to early morning low tides at Pillar Point. There's something like so amazing and beautiful about being like the first people out. And I noticed someone asked that in the Q&A also. Like, we definitely see different things when you're out there at an early morning low tide, for sure. Just because nothing's been sitting out in the sun all day yet. So it's really hard to think of one moment where like it's so beautiful. But in terms of like, I can think of moments where we found interesting things. So I'll talk about with Snapshot CalCoast last year. We took some volunteers out to Frenchman's Reef, which is kind of in between the Fitzgerald Marine Reserve, although it's part of the Marine Reserve, but it's the reef in between the traditional Fitzgerald one and Pillar Point, kind of north of Ross's Cove. And we were out there with some volunteers, and our friend Cynthia, you know, she was just like, whoa, look at this thing that I found, and called us over, and she found just the head of a wolf eel. And the inside was full of like snails and isopods that were eating it. And it had these amazing bright pink teeth. It was just like one of the most incredible things. It's the only time we've ever seen that out in the tide pools. And we just sat there like forever trying to figure out like, why is it just a head? I mean, it took us months. Like, why is it just a head? Why does it have pink teeth? Like, we started talking to our ichthyology collection manager at the academy, and like, you know, went and looked at like purple otter skulls and stuff like that. And it was just kind of this amazing mystery that like unraveled over months. But it was such a fun find, this crazy head of a wolf eel with amazing pink teeth out at Frenchman's Reef. One of the things that stick in my mind. I don't know, Rebecca?
Oh my gosh, it's so hard to think of something, but I agree. Like, they're just like the feeling of being in the tide pools and with a small group of people and just experiencing a place that's so special and magical, like as you're walking out and you can see the tide is low and you're like, what am I going to find today? Like that, just like that anticipation feeling is really, really great. I was thinking about maybe the last time I took my daughter tide pooling. She's 8 now, but this might have been a couple of years ago. She found an octopus, actually, like a small intertidal octopus. And we didn't make a lot of noise and we didn't call a lot of people over. We were just quiet and still and watched it. And we saw it use 2 of its tentacles like chopsticks and grab a shrimp and then eat it. And it was amazing. And she talks about it to this day, about the octopus that like caught the shrimp. And I mean, I have hundreds of those stories, but that one was just like extra special because it was us together. And because I'm in the tide pools a lot for work and not with my family, it's really nice to have those moments with my family in the tide pool.
I'm remiss here if I don't follow up with a great question from Peter R., who also saw an octopus once and wants to know, is that common in the tide pools?
Well, Alison is like the octopus whisperer, so we should let Alison
This is totally anecdotal. And like, again, as more people collect data on other places along the coast, we will see if this is actually true. But at Pillar Point, I would say that like, you know, we would probably find an octopus once every couple months. Again, they tend to kind of hide. And so I'm sure they're out there and they're harder to find. But yeah, for whatever reason, I usually had a pretty good knack for finding them. But I personally have not observed an octopus at Pillar Point since 2018. And so I think there's something going on with octopus. It's the Eastern Pacific red octopus that we find mostly in our tide pools. They stay small, they don't get big. But generally, yes, you can find them normally, but I think and I'll be interested to see if it's just at Pillar Point or if other people, if we're not picking up octopus observations along other places on the coast as well.
We have a question from Karen Moyes, who wanted to know about kelp. And I wonder if you can see in your intertidal observations any signs. As people may know, if you've seen the cover in the new Bay Nature, there's been a decline in kelp forests around much of Northern California. Do you see any signs of that on the beach in the intertidal?
This is a really interesting question. And this is another thing. Kelps are another thing that make Pillar Point actually really unique, a really unique place. Actually, along the San Mateo coast, they've seen an increase in bull kelp, right, which is the kelp that is featured in the story, and that there's been a huge decline on the north coast. But along San Mateo County, there's been an increase. And we do see recruitment into the intertidal. That means young plants or young seaweeds like trying to make their way into the intertidal. They don't really establish, but we do see that regularly. But at Pillar Point, there's actually a giant kelp bed, which is Macrocystis, which is the kelp that's more associated with the Monterey Bay, or further south in Southern California and the Channel Islands. So there's an intertidal kelp bed at Pillar Point, which is really remarkable. And I don't think there are many other places that you can find it. But again, anecdotally, we have seen a decline in the kind of heft of that kelp bed. It's still there, but it's much smaller than it has been in the past. And I don't know, there has also been a decline in giant kelp further south. So, you know, kelp's having a hard time, lots of places, but we have seen definitely some changes in abundance of the kelps at Pillar Point and other places. And this is another place that, like, if you're walking along the beach, it doesn't have to be a low tide, but if you see kelp, if you see rack on the beach, like kelp that's washed up, take a picture and upload it to iNaturalist. Like, this really helps us understand the dynamics of where kelp is washing up, how big it is, and you know, we can use these data to help understand what's happening offshore, because not everyone can dive, not everyone can do the amazing work of Reef Check on the North Coast, who are really looking at what's happening to the kelp underwater, but most of us who are near the beach can take pictures of things that we see washed up.
Yeah, it's a really interesting. We have a lot of people who've asked too, and along the same lines, you mentioned the sea stars a little bit. What have you seen this year? What do you see this year in terms of sea star wasting?
Yeah, you know, like Rebecca was saying at our Pillar Point slides, generally we've seen that those ochre stars seem to have recovered pretty well. You can definitely still find stars with wasting. I was out on the Sonoma coast, you know, just a few weeks ago, and I definitely saw one or two that definitely have signs of wasting. But the idea is that every generation you're going to have fewer stars that are quite as susceptible to the wasting disease. And so you can still see it out there, but on the whole, ochre stars seem to have recovered pretty well. And some stars that we have along our coast didn't seem to really be affected by it at all. There are a couple species, though, interestingly, that seem to be kind of locally extirpated because of the wasting disease. One of them is sunflower stars, which are the stars that have 22 arms, and they generally move pretty fast through channels in the tide pools. We haven't seen one of those since 2013. Right before the wasting disease hit. It was really the hardest in 2013 and 2014. So sunflower stars, people are not really finding them on our coast anymore. And then pink star, giant pink stars, Pisaster brevis minus. So it's a different species of Pisaster than the ochre stars. That's another species that we used to see fairly regularly. They were never abundant, but we would see them relatively commonly, one or two at Pillar Point, and that's another species that we haven't seen out there since 2013. I found them on the Sonoma coast initially in like 2015, 2016, but I also haven't seen them on the Sonoma coast there for a few years either. So while some species seem to be recovering, a couple others seem to be hit pretty hard along at least our section of coast.
Yeah, Rebecca, anything to add? Or?
I just add that there's some other interesting dynamics with Leptosterius, which is the 6-rayed sea star, which is really common at some places and then not common other places, and was hit by the wasting, but it was a little delayed. And it was a little harder to figure out because it's so patchy, but Dr. Sarah Cohen and her students at San Francisco State and the EO Center have done some really, really great work to understand this species. But one of the cool things about this species is when you're looking for nudibranchs and you're being really still, it's a very small starfish. When you're sitting still and looking for nudibranchs or other things that maybe seem a little more charismatic, you can often see Leptosterius and another small star called Henricea. So they kind of live in the same place. So it's fun to see them. And I've been seeing them a little bit more recently. So.
Okay, I think we have time for one more question. This is an interesting one from Deanna DiPietro. Does it help to record observations of common species?
Yes, yes, please record common species. So when we have a list of species that we ask people to look for Snapshot Cal Coast. I mean, we really want people to look at everything, but we usually have a most wanted species list, which is usually things that we know are shifting ranges, like moving north, or invasive species. But a few years ago, maybe 2 years ago, we started asking people to take pictures of really common things and reminding them to take pictures of barnacles and limpets and anemones, because we're using those data to say maybe this thing isn't here. And so we know that there are barnacles at most beaches, if not all rocky intertidal beaches. So we're really asking people to kind of have a little checklist, and we have a checklist available. It's on our website to remind you to take pictures of everything. You don't have to take pictures of every single individual you see, but like every time you see a different species or you think something is different. So kind of every species that you see. And it's okay if you do some twice, like don't worry about it. We're not counting abundance, we're just saying this was here. So yeah, I don't know, Alison, if you have anything else to add.
Yeah, no, definitely take photos of common species. And I know I saw other people just asking like, can you participate from anywhere? Yeah, anywhere along the California coast, just get out with iNaturalist. Take photos of anything that you see. I mean, even if you only have 5 minutes to be out there and take some photos, that's still data that we can use for sure. So anywhere along the California coast, we'd love to see your iNaturalist observations. And generally, both of us are pretty good about watching California coast observations show up on iNaturalist, and we can help ID the things that you saw too, if you weren't sure what they were. So that's another way to interact with us a little bit more if you're not in the Bay Area.
Okay, well, I just want to close. Maybe actually, can we remind people the websites we've mentioned? How can they find the citizen science program at the Cal Academy?
The best thing to do is just to go to calacademy.org, and then if you just search for citizen science, it should take you to our citizen science webpage that has information about Snapshot Cal Coast. It has the email that you can use to tell us you want to be on the Pillar Point list. You can sign up for our newsletter too. We don't just work in the tide pools. We actually do some terrestrial biodiversity work as well. So if you're just like, yeah, I'm not really feeling like up to slippery rocks and early mornings, then sign up for our newsletter and hear when our bio blitzes are happening in parks and stuff like that.
Great. Well, I want to thank everybody for coming and hanging out with us this afternoon. But thank you, Rebecca. Thank you, Allison, for sharing this with us, for sharing the California coast with everybody through iNaturalist and through Snapshot all the time. Thank you guys very much for being here. Thank you to our audience for some great questions and for being here with us. Again, check out Bay Nature and check out the California Academy of Sciences Citizen Science team. And we will see you next time.
Thanks, everyone.
Thanks, everybody. Bye.
Bye. Nice one.
All right.